So thank you, Micha, for a very nice introduction. And thank you, Michael, for inviting me. So Michael and I, we know each other for a long, long time. Too long, well, too old, right? Uh, so it's a it's a diverse audience. So I thought about what to talk about, but then I couldn't decide. So then I decided to put everything together. I will tell you three stories, but since it's three stories, each one will be have to be short. So three short stories, just trying to squeeze in so that everyone hopefully can at least catch a little bit of he or she is interested in. I don't have a better title because I put three things unrelated to each other, so I use a very arbitrary title. I guess the common theme is that computation, right? It's the Center for Computational Biology. Most of them are computation here. And as Micha said, I'm a physicist, so it's a kind of angle from a physicist point of view to do computational biology. Okay, so so the three stories basically one is bacteria. It's a very old phenomena, Diauxie versus co-utilization. So we solved this problem in terms of optimization. And then a very hot stem cell reprogramming problem. We built a mathematical model to understand the reprogramming and also propose some new protocol for reprogramming. And finally, something we did very recently, we use deep learning to try to reverse engineer genetic networks. Okay, so the first problem, bacteria. So that's my breakfast this morning. Not exactly. So when you are presented with a bunch of food, right, what's your choice? Do you eat them simultaneously or you eat one after another? This sounds like a game, but Jacques Monod did this exact same thing for bacteria. So he feed the bacteria with combinations of carbon sources or sugars. And then he found that in some combinations, he got a single growth exponential curve. And this is OD, this is bacterial density, this is time. It's a single exponential. In some other combinations, he saw two phases of growth, exponential and stop growing, and another exponential. Okay, and then he did a very careful analysis. He's really, one of the founding fathers of quantitative biology. He did quantitative analysis, and then concluded that in this case, the bacteria is eating one carbon source first. After one source is exhausted, they started to eat the other. Okay, and then he coined this phenomena Diauxie, sequentially utilized two carbon sources. And in this case, the bacteria just eat the two carbon sources at the same time, simultaneously. For example, if you present them with glucose and the pyruvate, so this is called simultaneously utilized. Okay, and this is before the Second World War, and this is from his PhD thesis. And then he was a very active participant of the French Resistance. And after the war, he came back to this problem, but by then molecular biology already was born, and he looked at the underlying genetic regulation for this phenomena, Diauxie. So he asked why bacteria can utilize one carbon sources first. What's the mechanism? And then, as many of you know, with Jacob, they discovered lac-operon, right? So essentially, there's a repressor called a lac repressor, usually represses the enzyme to digest lactose. So if you give the bacteria glucose and lactose, bacteria likes the glucose. They will eat glucose first. They digest glucose first. Why don't they digest lactose? Because the enzyme digesting lactose has been repressed by lac-operon. Only when the glucose is exhausted and lactose are present, this repressor is being released. And then the enzyme digesting lactose is being expressed. So this is called catabolite repression. And this is end logic. Only when you have lactose and no glucose, and this genetic regulation is turned on. And this is the first example of genetic regulation. And for that, he got a Nobel Prize, shared with Jacob, right? It's a very famous Lake operon. Okay, and later on, people found that this phenomena is very universal, namely sometimes in some combinations, they co-utilize, in some other combinations, they eat one first and then the second. And in bacteria and even in some eukaryotes like yeast. Okay, so if we call this phenomena what? So what happened? So what we see. And we call this a mechanism how. How they achieve this? Just by repression, catabolite repression. Okay, in this case, repress the enzyme digesting the second source, unfavorable source, okay, by the genetic repression. And I won't ask the question, why? Why, bacteria sometimes they eat two simultaneously, and sometimes they eat one first? Is there any reason? Okay, so the bacteria eat carbon sources or nutrients to metabolize them to make proteins and nucleic acids, right? To reproduce, to produce biomass. So this is their metabolic network, somewhat simplified, but you can see the main thing, the glycolysis, the TCA cycle here, and the carbon sources enter the metabolic network from different points. And all these green ones, these are different carbon sources. They enter from the upstream of the glycolysis, converging onto this point. And another kind of carbon sources we call B-type. They enter downstream of the glycolysis, okay, essentially. And this is somewhat simplified. And then the arrow here means there's a transporter, maybe some enzymes also, blah, 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 several steps and converge to this point, and all these arrows. So this is somewhat coarse-grained network, but the topology is essentially like this. Okay, then experimentally, people found that if you put two carbon sources from the group A, like two green carbon sources, you see Diauxie. They eat one source first, and then the second. So they have a preference. If you feed them with 1A and 1B, they eat them together, co-utilization. So that's the phenomena. That's what people found, right? And Jack Monod found that if you put two A's and then one of the enzymes digesting one will be repressed, right? Remember. Yeah, please.
So in this diagram, can I see some kind of interference between the different sources, or
What do you mean by interference?
Competing for something in that?
Okay, so you already guessed the answer, right? Right. For Diauxie, exactly, they're competing to enter this the common point. So they will use the one which will take the least resources. Okay, that's part of the solution. Okay, so first let's look at group A. If we present two sources from group A, and how they are going to reach all this we call the precursor pools, like all these metabolites, right? They will produce amino acids, different amino acids. See, all these different groups of amino acids are produced by different precursors from different points. Okay, all these orange squares, they are amino acids or nucleic acids to produce biomass. So as Mitch already guessed, there's a competition if you use the combination of two A's, because they all have to converge to this common point and then be diverted into different pools. And topology is here. So they have to converge to this endpoint, that's here, right? And so you have carbon source outside the cell and then being transported in and being metabolized several steps, typically many steps, and then converge to this common point and then being diverted. So that's a topology. So we just have to solve that topology to ask what's the reason they want to use one versus the other, right? Why don't they use both simultaneously? Wouldn't it be better? Okay, then we can define the quantity. Yes.
Is it always strictly efficiency of conversion, or do other factors such as troublesome waste products ever factor into what the ordering of input?
There are some complications, yeah. So what I'm presenting you is sort of the zeroth order approximation. It's probably 80% correct. Okay, you can, yes. So so then we can define some enzyme efficiency, defined as the total carbon flux delivered divided by the total cost. These are the enzyme mass. Okay. So if I want to ask what's the efficiency for the carbon flux going to the M from all these branches, then it's just the total flux, which is flux from A1 to M, flux A2 to M, this flux plus this flux, divided by all the enzymes I use in this branch, and all the enzymes I use from this branch. So that's the efficiency to deliver these carbon sources to this metabolite, okay? And then you can calculate each branch, the efficiency, namely the flux in this branch divided by the cost of enzymes in this branch. And this can be calculated just by Michaelis-Menten formula. And all these constants are the Km and Kcats of all these intermediate enzymes, okay? So if you do this, and then you maximize the efficiency, and as Mitchell already guessed, it is most efficient if you use the most efficient one branch, okay? It's simply the maximization. So which means that if the efficiency, if this quantity, if this pathway is higher, it's more efficient, you better shut off all the enzymes here. You should devote all your resources, protein resources, to express enzymes in this branch. Okay, so very reasonable, right? Very, very simple, actually.
I guess part of this is that you're assuming that downstream of M, there's no further memory of what the particular carbon source was.
There is no further memory. This is just a chemical. Yeah, it's a chemical. Okay. And then this, if you look into this a little bit more carefully, this formula, I said this is a Kcats and Kms, except there's a quantity here which depends on the concentration of the nutrients. This is the first step. This is transporting the nutrients into the cell. Of course, it will depend on the concentration, as you might guess. So for higher concentration, the efficiency will be higher. For lower concentration, the efficiency will be lower. Okay, so the efficiency of the pathways depend not only on the pathway itself, but also on the concentration of the carbon sources. It's simply because Michaelis-Menten's equation, there's a substrate concentration there. Okay, then we can go back to the Monod's problem. We presented with them 2A carbon sources, maybe glucose and lactose, and then they will eat the carbon source with high efficiency first. For example, this is glucose, and you can use less enzyme resource to digest. They will eat this one first and not express any enzyme here. Okay, so that's this growth curve. And then after this, the carbon source being exhausted, or more strictly, after this two efficiency being balanced exactly equal, okay, and then they will switch to the second one. Okay, they will switch to the second one. Okay, then we can ask, what's the turning point? When should they turn? Did they really exhaust everything and then start to turn? As we said, this efficiency depends on the concentration, right? It depends on the concentration of the second. If the concentration of the secondary carbon source is very high, they should turn it earlier, right? Okay, so we can calculate the turning point. You can calculate this, and this is a formula. So the concentration of the second less preferred carbon source is higher, I should turn earlier. I should not eat to the last bit. I should switch earlier. And that's the formula. And then I can do experiments. Okay, but before Mike Springer at Harvard, they already did experiments, not for this purpose, but for some other purposes. So they grew yeast on 96 plate, and they say on each well, there's a galactose concentration gradually increasing on the x-axis, and the glucose concentration increasing on the y-axis. And then they ask, is the yeast eating glucose or galactose, right? There's a reporter gene to report that. And then they found that in here, the yeast is eating galactose because the enzyme digesting galactose is being turned on here, okay? And here they are eating glucose. And this is precisely what we predicted. So switching point depending on the concentration of the second source. So if you label this 50% cell switched as a switching point, and then our prediction would be this curve. So it fits almost perfectly. Okay, so we thought we understand why they want to use one versus another, because it's more efficient, right? So this is Diauxie. But why co-utilization? Why sometimes they use both simultaneously. In this case, indeed, they grow faster than eating on any one of them. Okay, so which means they are really making advantage of the two carbon sources. Let's look at the metabolic network again. This is group A sources, group B sources. Okay, and the metabolites entering the precursor pools from different points. As you can see, for example, these metabolites will make these amino acids, these metabolites will make lipids, and this one will make these amino acids. In this case, the topology is very different if we use one from A and one from B. So for example, if we use one A source and one B source, in this case is a pyruvate, it's a little complicated, but the topology is like this. So the A source will also enter that endpoint, if you remember. They will converge. Many A sources will converge to this point. But the B source, carbon source, they enter the metabolic network from the lower part of the glycolysis. They have some shortcut to some other pools, right? So it's also very reasonable. You can simplify this topology like this. So basically, you have more than one pools, and the A source can reach both pools, no problem. And the B source can also reach both pools. But maybe A can be, it has a shorter path to pool 1, and the B has a shorter path to pool 2. So that's essentially the reason why they want to use both. So everything's very simple. You can do the same calculation, right? In this case, if you have A and B, would they pull A from A or pull B or pull 2 from B? You can calculate the efficiency and then compare, and find out that in what cases what pool will be supplied by A or B. So essentially, there's a large range of parameters where A should supply pool 1 and B should supply pool 2. Simply because maybe the intermediates can be very long. Okay, it becomes less efficient. And you can do all this calculation, use the enzyme parameters, which is lots of them are known for E. coli. You can do this. Okay, but this sounds too easy and too trivial. Can we make predictions? That's the point. Okay, so you say they should be co-utilized simply because A is closer to some pools and B maybe is closer to some other pools that we just take them simultaneously. It's more efficient. Can you make predictions? So if I supply the E. coli with one A source and one B source, can I predict which of the amino acids will be from B source and which of the amino acids will be from A source, right? And then you can do the calculation. So for example, if I give them glucose and pyruvate, and this is glucose, and they enter the metabolic network from the upper part, and this is pyruvate, they enter it from this part, and I do the calculation. So for each pool of amino acids, I calculate the enzyme efficiency, right? Is it more efficient from here, or is it more efficient from there? And then I decide it's from A or B. And then you can show that from this, all these amino acids will be supplied by A, by glucose. And all this one will be supplied by pyruvate. And simply because the TCA cycle is turning, and these amino acids, these two groups will be supplied by both. And we can calculate the percentage. 60% maybe is from glucose, and then 40% of carbon is from pyruvate. And here's a different percentage. So you can do all this calculation. And then you can use carbon 13 labeling to verify. So essentially, you feed them with carbon 13 glucose, okay, and after a while you measure, use mass spec to measure which amino acids has carbon 13 and which ones don't have carbon 13. Yeah, please.
So when you say you're looking at these statistics, do they apply does an individual bacteria do both processes at once, or is it population?
That's individual. All here is individual bacteria. Yeah.
Making these quantitative predictions, how many free parameters does your model have that you're fitting, and how many are coming from literature values?
The parameters, most of the parameters are coming from literature, but some we don't know, so we'll have to make some guesses. But as you can see, these are actually quite robust prediction. It's topology, essentially. Okay, it's a topology. It's a topology of metabolic network. So although maybe some enzymes, the parameters are not so clear, but within the range, the prediction is the same.
It's actually a little surprising to me. So Michaelis method kinetics are the standard in the field.
That's right.
But they're based on approximations that were made in the 1920s.
Yeah.
To linearize things that now we wouldn't linearize. So it's kind of surprising to me that these approximations work this well.
Yeah, but on the other hand, you can see that the topology also means a lot, right? Okay, so I wouldn't bore you that we can make predictions for any combinations of A sources with B sources, and then compare with experiments, and they agree pretty well. The hollow bars are model prediction, and the solid colors are the experiments. These are different amino acids. This is a percentage from A source or B source. Okay, very they agree quantitatively. Okay, so for this story, it's actually very simple. It's essentially this simplified topology. You can understand everything. You have different sources, carbon sources, but the topology of the metabolic network is such that all the A sources converge on the common point, and then they are being diverted into different pools. And the B source can enter from different point, and they can take a shortcut to supply some pools. Okay, So that's why when you have two A's, they only take one. They take the most efficient one, either this one or this one. When you have A and B, they take both. And if you have two B's, and very often they also take both. Okay.
Sounds like you should be able to take a mixture. So you take one A, you take a mixture of the B's, and you maximize the total amount of biomass.
Yeah, one can do that too. Yeah, to get the maximum growth rate. Yeah. Okay, your question?
Oh, I don't know if it's the same, but in the first one, once you reach that balance point, wouldn't you want to take both?
Oh, yeah, very good question. So you must be a mathematician. The bacteria actually, it's a bistable switch. Yeah, some people ask when they reach this balance point, they will eat another one, and maybe the concentration decrease, and we eat this one and this one and this one. No, the bacteria wouldn't play this game because it's too costly for them to switch enzymes. They just switch once. So it's a bistable switch. Okay. And then what time to switch? There's some stochasticity. Yeah?
Okay, so when Jacob and Monod, they did their experiment, what came out from that was they were able to predict the lag operon, and then for you, when you did this, so what's the next step after you're able to predict this thing? Because it seemed like you mapped everything from a metabolic network that is already known.
Yeah. So okay. So we ask different questions, right? We ask why they do this. Why sometimes they eat both, sometimes they eat one, and we think it's from the efficiency, the growth efficiency. They can grow more efficiently, which means they grow faster. Okay. If they can grow faster, then they always find a way by evolution to do this, right? Like they evolved like operon, and then when you have A and B, then they just express both enzymes. There's no repression. So all these are the mechanisms. So here we are not concerned with the mechanisms. They are of course interesting mechanisms. How do you realize this? Sometimes eat one first, sometimes eat both, right? Yeah.
So given your model with all its parameters making good predictions, if the system is changed, does your model continue to make good predictions? For example, let's say you have a strain of bacterium with a mutation in one of the enzymes that reduces the catalytic efficiency. Did you predict?
I don't think we can make good predictions because all this is by evolution. So the bacteria evolved a strategy like this. So if you mess them up, like you change the enzyme or change the parameters, the bacteria doesn't know what to do. Yeah, I don't think they will calculate again and Yeah. There was a question there? No. Okay, so let me switch gears. Yes.
So what I didn't understand is that you were looking at a population of bacteria.
Yeah, but this is why you can think this is a single cell. Yeah, this is a single cell. This is a strategy of single cell. But for the population, the only difference is that when do they switch? Are they exactly become, this seesaw become balanced, switch, or they switch a little earlier or a little later? And then if you observe within the population, each individual, they will show individuality. Some will switch earlier, some will switch later, like bad hatching. Yeah. There's no cooperativity. Yeah, they just do them some randomness. Yeah. Okay, so stem cells. So each one of us, as you know, is from a single stem cell and then developed into several hundred different cell types. And the biologists are interested in the transformation between different cell types, or better, from differentiated cell back to a stem cell. And this has many potential applications, right? Okay, and actually in the early, I think most people know this very famous Waddington landscape. The biologist, developmental biologist, Waddington, is speculated that why developmental process is so robust It's because you can imagine there's a ball rolling down the hill, and all these grooves are very deep so that the ball will not make mistakes. And different paths are different cell types. So this is a stem cell. They will develop into different cell types. And then he further speculated, so that's very interesting. What makes this landscape? And he said, well, these are the genes, and they have the interactions, so that made this landscape. Okay, now we have all this data ready to reproduce this Waddington landscape to understand development and the stem cell reprogramming. So maybe I don't need the introduction here. So this gene expression is high dimensional space. And then you can write down the, for gene expression, there are different fixed points, right? If you write down the simplest, a autoregulation of a gene will activate its own expression. We call this positive feedback loop. And you can write down differential equations. This is a activation term is squared because you have a dimer here. And this is a decay term. And for this, you can have bistability. You can have two fixed points, stable. Either this gene is not expressed at all, or they are expressed at a high concentration, right? So this is just to say that even a single gene, we say autoregulation, you can have two fixed points, right? Yeah, if you have two genes, then you can have four fixed points if they are not interacting, right? So every cell type is in fact one fixed point of the dynamic system. So that's what we thought. That's what many people thought. That's a view. It may or may not be correct, but we thought it's just a fixed point of the dynamic system. The dynamic system is the gene expression dynamics. It's just differential equations. Okay. So if my cell has two genes, I can make up four different cell types because I have four different patterns of expression, right? Stable. So if they're interacting, this can change. This can become only two fixed points, or maybe the repression, the mutual repression is not strong. I can have three fixed points. So I'm saying this is just this is a very, very sort of simple introduction of cell type corresponding to fixed points of the dynamic system. And the landscape can change, as you can see, by changing the interactions. Okay. Then the dream of the biologist many years ago was to transfer a differential cell, for example, fibroblast, into a stem cell. Okay. And with many tries, they succeeded in 2006 by a Japanese scientist, Yamanaka. He got a Nobel Prize for this. He tried many genes. Many genes are usually expressed in stem cell, and put them into a fibroblast cell. But eventually, he found that he can use only 4 genes to do it. So you can see this is a very low-dimensional system. So this is very encouraging. We have 30,000 genes in our cell. But you can use 4 genes, overexpress them in a fixed point, which is the tail, and then you can drag them into another fixed point, which is stem cell. Okay. So in this simple picture, so maybe this is a tail, this is a fibroblast cell, very stably, and you change 4 variables, you made them from here to here, stem cell. Okay, and then my colleague at the Peking University, he's asking the question, can I replace the so-called Yamanaka factor? Can I replace some of the genes by some other genes? Do I have to use this 4? I can use something else. And what he did is what biologists typically did. He didn't use this one, Octo4, and he tried many others by screen. And he found out that yes, indeed, he can replace one of the Yamanaka factors by one of the many other genes candidates. But to his surprise, that remember, if we want to transfer for example, the tail fibroblast to a stem cell, Yamanaka used 4, and now people know that these 2 are in fact to open the chromatin structure. Okay, they just flatten the landscape. And these 2 genes, other genes expressed in the stem cell. Okay, so you flatten the landscape, make the hills lower, and then you just drag them with the degrees of freedom of the stem cell, and you can drag it to the stem cell. So that's Yamanaka succeeded. And my colleague replaced one of them with other gene, and to his surprise, he can replace it not with another stem cell gene, like a red gene. He can replace it with blue gene. So he couldn't understand this. How can I, transform a blue cell to the red cell, but not using the red gene, but use a blue gene? It seems a contradiction. Anyway, so they did lots of gene expression data and then figured out some interaction between the genes. And then they came to us. They don't understand. They say, can you explain what's going on? So then we write down a dynamic model using their data and their intuition, their understanding about the relationship of the circuit. So it's essentially a 4 degrees of freedom. You have 2 important stem cell genes, and we know they are co-activating each other. They're enhancing each other. Okay. And then one gene is actually a group of genes representing one cell fate. This is a meso-endodermal, I think. And one blob of gene representing another cell fate, ectodermal. And we know they are repressing each other because you cannot be simultaneously a heart or liver. So they are either one of them. Okay. And if they are expressed, they will go back to repress the stem cell genes. And then it's interesting, the stem cell genes are activating these 2 genes in a very symmetric way. So that's the network we got, and then we can write down differential equation, not very complicated. And then you may also ask, you have so many parameters, how do you rationalize all these parameters? But again, I should say that parameters doesn't matter that much. It matters, but not that crucial. Okay, then you can solve this differential equation, or you can solve it numerically at least. And then indeed you can find there's 3 fixed points. One stem cell fixed point, namely these 2 genes are expressed, but these are repressed. Okay, so the fixed point is here. And each of these are corresponding to 2 differentiated cells. Either this one is expressed, everything is repressed, or this one is expressed, everything is repressed. Okay, so these are the same pictures. It's 3 values, 3 fixed points. Okay, and then from this picture, you can actually understand the development and reprogramming as a balance of seesaw. So the stem cell state, namely these 2 are expressed, are very balanced state. The 2 branches, they have equal force. Okay, they have equal force. They're sitting here in the middle. It's a symmetric state. It's stable, but symmetric. And then for the stem cell to commit into a differential cell, basically you tip the balance. You can either increase the expression of this one or increase the expression of this one. Doesn't matter. Just if this branch is more wins over this branch, then the balance will be tipped, and then will be the fixed point will change to this ME state. If you overexpress this branch, they will change to another differentiate cell. Okay, and then reprogramming this view. So that's development, tipping the balance. And reprogramming is just restore the balance, right? So originally, maybe you are sorry, I don't have this. So maybe originally you are here, if you restore the balance, then there's a chance you can go back. And that's what Yamanaka did. He didn't have this picture, of course. He just tried, and he succeeded. Basically, he overexpresses 2 stem cell genes, very intuitively correct, but in fact, it's restoring the balance of these 2 branches. Okay, so originally you have a differential cell, fibroblast, for example. You overexpress this 2, you restore the balance, and then there's a chance for you to go to the stem cell fixed point. So my colleague discovered you can replace this one by a gene in the differential cell, also go to the stem cell state. But that's also restore the balance. Okay, and he also found that you can do this way. Just by replace this stem cell gene with a differential cell gene, you can also get to the stem cell state. Okay, so this can explain all the experiments, but also make a very interesting prediction. To restore the balance, you don't have to use any of the stem cell genes. You can use the differentiated genes. So which means to go to this red cell, you don't have to use any genes in the red. Okay, so this was a big surprise to biologists. So which means in order to go to a fixed point expressed by these 2, you just let these 2 genes fight. Maybe a skin with neuron, let them fight. And if you're lucky, and you can go to stem cell state. And in the dynamic system, it's actually very simple. If you already differentiated here, you just drag them into the diagonal. You express both and release it, and you can go back to the stem cell state. So or like here. So that's a prediction, and then they did the experiment and succeeded. So they just used 2 differential cell genes, let them fight, and then get a stem cell, and then can grow a mouth out of it. Okay, so we call this a seesaw model of reprogramming. So it's basically restore the balance. It doesn't matter how you restore it. If you restore it, they have a chance to flow to this stem cell fixed point. Okay.
So how, it clearly isn't any cell type specific genes, right? Do you guys have a sense of what are the qualities of these cell type specific genes?
They are these ME genes, and I have the names. So if you are interested.
No, I'm just wondering, like, what is it that makes, right? Like, it's not any cell type, it's not any mesodermal, let's say, expressed gene, I'm assuming.
They are.
Right?
They are.
Any gene that's expressed in the mesoderm and any gene that's expressed in the ectoderm together? Or does it have to be?
Yes, you just use 1 gene expressed here and 1 gene expressed here.
So any random.
Oh, you mean any or some?
Yeah.
Okay, that's good question. That's good. That's
it depends on where they are in the pathway.
that's exactly. That's okay. Yes. Yeah, not any. I don't think it's any. Some. It's group.
Do you have any sort of predictions from your model of what are the properties of those genes that?
Okay, that's a very good question. So you're asking a high resolution model, right? We don't have it. Yeah. So ours is lower resolution model. Yeah. Exactly. So if you really figure out the pathways, you should be able to make more predictions. Yeah.
So the obvious reason that we want to make pluripotent cells is for regenerative medicine, right? We're trying to turn cells into types that. Can you make predictions about how to get from a differentiated type to a differentiated type?
That's actually easy. You just express genes here. They will be dragged. Yeah, and people know this. Yeah, it works. Yeah, but as Olga pointed out, it works for some genes, but maybe not all the genes. Yeah, to really understand what genes work the best, you have to study the pathway in more detail.
This may be a naive question, but are all pluripotent cells created equal? Like when you drag them back, is it truly the same? You know what I mean? Like
I know what you mean.
But is it truly exactly equally pluripotent?
No. Yeah, I think they have hierarchies. Yeah, right. Yeah, that's the short answer. A more detailed answer, actually, I'm not qualified to talk. Yeah, that's right. Yeah, I think
But if you expand your model, you might be able to actually predict.
that. Yeah,
right? Like
if I including more pathways. Yeah.
So in the network, there's no way that I see that increasing the lower genes causes the upper genes to increase.
Yes, in the network, yeah, first glance, yeah, there's no way you can see. But mathematically, it's actually easy to understand. Pardon me?
Are there missing arcs in the model?
No. There's no missing parts in the model. From this model, you can, yeah, if you do the calculation, you can. No, if you start here, if you start from here, you express any genes from the branch as long as they are balanced. Okay, balanced meaning that you should use both. But what are the quantities? That's a more quantitative prediction, which we don't have. But as long as these two branches are balanced, then you have a large chance to go back to this stem cell state. It's in the network. There's no missing path, missing component. You mean here? Yeah. See, this is the sure. When this is expressed, you will express this. When this is expressed, you will express it. But you can also see they have this cross activation and also cross inhibition. So when the when both are expressed, I think the most clear picture is here. You just drag it. You let them fight, and then there will be out of this this well, and then they either flow back to the nearest fixed point, or they can flow to this fixed point. You let them fight. So the cell was sitting here, but this is his enemy. And this enemy will drag it back out of this comfort zone, and then it can flow. It's like here. If you drag it into the right position, then it can flow here. Of course, you can drag it to here, and then maybe it will flow back to here. But there's a range of this force fighting. Then it will flow to the third fixed point. Yeah.
So in the first part of this talk, you showed the example of the lac operon and how
Right.
how genes are regulated by metabolites and interaction with the environment. Here we've got 4 genes that are being treated like they're interacting in isolation.
Right.
Does the environment enter into this model somehow, or both genetic background and the other factors that can affect gene expression?
To some extent, yes. So here is I studied the isolated module, and they are sitting in a much bigger genetic background. Of course, they will be influenced by the other genes. But on the other hand, I can already understand some features even I don't consider the environment. Or the environment, they are fine-tuning the system, right? So if I want to make more precise predictions, like what's really the gene dosage of this gene and that gene, what's the probability I will flow to this stem cell fixed point, then I probably need all these other factors. But here is sort of a very coarse-grained. So after we made this prediction, they tried, and they didn't try. They didn't listen to us. That okay, I use this expression of 1, this expression of 1, and then maybe or 10. They just tried 10 to 3, 3 to 10, 5 to 5, right? As long as you express both, and there's a chance to go there. Okay. So the last story is the deep learning. I guess many people will be interested in, but here's the application of deep learning, not the theory. So so one thing in biology is really mapping the genotype to phenotype, right? Biologists spend a lot of time doing that, and essentially you have bunch of genes and proteins interacting, and you have a network, and you have gene expression, and then you have some phenotype. So that's a flow of information. But in fact, this is being solved traditionally inversely. So biologists are making mutants, genetics, knockouts, and then to figure out what genes are interacting with other genes. And then by biochemistry and molecular biology to see the mechanism. For example, like operon, is this transcription regulation, or is this enzymatic regulation, or is this kinase? So the flow goes that way. And now we have more and more data. Here, gene expression, even gene expression dynamics, which they change with time, right? And then there are many statistical methods, like bioinformatical methods, to try to go from gene expression to network. But as you know, most of this inference are very powerful to infer the correlation, like clustering, for example. Less powerful to infer the causation, like which gene really is regulating which one, rather than just they co-express together. Okay, so if you look at it, it's really, again, this genetic network is an underlying dynamic system, right? So genes at time t+1 or t plus delta t is the previous time plus some increment, which is dependent on all the other genes. You can view this as differential equation or a Markov process or some discrete updating process. It doesn't matter. But it's a dynamic system underneath. So it's that this dynamic system from this network to generate this gene expression dynamics and then phenotype. So we ask, can we use this data to infer this network? So what we mean is that infer this, we call F function. Okay. So I don't have any specific constraints for this functional form. So it's unrestricted and no parameter fitting. I just want to infer this F function as accurate as I can so that I can reproduce this dynamic system. Can we do that? Machine learning. Okay, DNN, of you know, can do magic things. And we want to read the genes at time t and tell me the increment of gene I, okay, at next time step. So first, let's do a very simple example, adaptation. So adaptation is a very ubiquitous function in biology. Basically, you have input, and if the input suddenly changes, and this is output, your output should respond, okay? But if the change persists, you should go back. So this is called adaptation. If you go back exactly as before the change, this is called perfect adaptation. So this is a very, very ubiquitous function. So can we say, given this task, can we infer the genetic network to perform this task? It turns out we can do that. So we use a deep learning network, DNN, as I said before. So I input the genes time t and also the input. So I have 3 variables here. And through a black box, I generate F1 and F2, which is the increment of gene 1 and gene 2, right? And I ask this, I iterate this, ask them to produce a target pattern for one gene, for example, gene 1, like this. It turns out it's very easy to train. Very fast converges, can produce this gene expression at gene 1, very close to the target pattern. And gene 2, I don't have any constraint. And sometimes, somehow they just have this expression pattern as I change the input, okay? You will not be surprised, right? DNN can do anything. Of course, they can do this. So what's interesting is that we want to open the black box. I want to solve this problem to see how the genes are connected, right? And this is usually impossible because In DNA, everything is connected to everything else. It's a huge number of parameters here. But there are a few tricks to do that. For this simple system, one can just plot the F function. Okay, I have 3 variables, input, gene 1, gene 2, and then I can fix, after I can finish this task, perform this task, I can fix the concentration of gene 1 and gene 2 and change my input. Okay, I fix the concentration of the 2 and change my input, and I see how F1 and F2 changes. I can see F1 goes up with my input, F2 goes down with my input. So which means my input is activating G1, goes up, right? And inhibiting G2 because it goes down. You can do the same thing, fix 2 other variables and change 1. You can figure out the relationship between the 3 variables. So that's the network you get after by plotting this F function. And that's precisely the network we know that it can produce perfect adaptation. Okay, so that's very simple. So another trick, which is very useful when the F function is not so easy to plot, when you have many variables, it's high dimension, you cannot plot it. How do you do it? Okay, one way to do it is that you can decompose this network into many networks. Each one will be responsible for one F function. Okay, so I have 3 variables. I have one F function, and this is something I should learn, right? Okay, and then after training, I can knock out any of the specific links. So I can knock out the regulation from gene 1 to itself, from blue to blue, and see the phenotype. And this is exactly how biologists are trying to figure out the network. So I knock out the G1 to itself. That's here. This is a Y-type adaptation, right? After knockout, the G1 goes up. So which means they repress itself. After knocking out the repress, this go up, right? So I know that G1 will repress itself. I do this for all the other knockouts. And then you figure out the network, which is exactly the same as the first method. Okay, this is simple, right? People look puzzled. Okay, so the idea is that after you train the network, you can do in-silico mutagenesis and figure out the relationship of these genes. So you know what is positive, where is positive regulation, where is negative regulation. Okay, I don't have much time, but let me go fast to Drosophila. So this is a very simple case. Then we just do it for real data, very complicated network. So Drosophila, very famous for development, is a cascade of gene expression, right? The pattern formation. So we're concentrating on this gap gene expression, which is formed by the signal for the morphogenes, right? So if you have morphogene, here we have 3 inputs, or 4, we actually only pick 4 outputs, and there's a very complicated network. And then this is the kind of data we use to train the system. So morphogene, people can measure their expression from the anterior, posterior, and then the 4 gap genes, you can do the immunoprecipitation, immunostaining, to measure their expression in space and in time, because you do them at different stage. Okay. So you have this dynamic data to train the network. It's very similar idea, except here is you have a spatial component. You have 4 gap genes instead of 2, and you have 3 inputs, and then you have a neural network in between. You iterate this. You train the F function. Okay. And it's also, training is very fast. You can fit the training data. There's the 7 frames of the dynamics. We just use wild type, essential wild type to train it. And then can predict, is post-dict, right? With many, many mutants pattern from the literature. You can compare with the, after training, you can predict all these mutants. It fits very well. So here's a summary of the fitting. If they fit very well, they should be on the diagonal axis. If some feature appeared on the model prediction, but not on the data, like here, I think that there will be, yeah, and then there will be on this either vertical axis, if the vice versa, will be the horizontal axis. So out of all the mutants, about 90% can align in the diagonals. So very powerful prediction. No model before have this kind of prediction accuracy. Yeah.
So what happens if you put in a gene that is not part of that network?
In training?
Yeah, just like in the model and in training.
Okay. Yeah, that's a very interesting question. So you can put extra gene that doesn't do anything. Yeah, that doesn't do anything. You can even delete a gene and then just train the 3 remaining ones. They do also reasonably well. So the DNA is somehow they find a solution. Okay, and this again is not so well. I think this is surprising. We're very surprised because they can predict all these mutants so well. They can predict some very weird mutants, triple mutants. I don't have time to tell you. They can all predict. So now we trust this learned dynamic system, this F function, because it can make so many predictions. Okay, and now we want to open the black box. That's the way I showed before. We have 2 tricks to open it, right? One is to do the in-silico mutagenesis. For example, I can knock out this blue gene to this what is this? To the blue gene itself. Okay, I can knock out any of the regulation, red to red or red to other, and see the phenotype. So, for example, if I knock out the giant to this KNI regulation, one of the links I knock out, so you can see this the red expression pattern, which is this red, invades into this giant region. So which means here in Y type, the giant is actually repressing it, so to prevent it to invading into this region, so that you can conclude giant represses the KNI. Okay, so you do the same thing for all the links. So you can infer the network like this. You can compare with the best knowledge of biology is this. Essentially, the core structure is the same. Okay, and there are some redundancies. The core structure is conserved, it's the same core structure, but there's a redundancy. For example, you see there's a positive interaction here, and there's a no positive interaction. Biology said, no, we didn't find a positive interaction here. Why? Oh, because you can see we have negative and negative. So double negative is actually a positive. Okay, so the DNN, they don't care. They just say, okay, this can be positive here. Although you have a double negative already here, they say it doesn't hurt to have a positive there. So you have this redundancy or equivalency, but otherwise it's essentially the same. Okay, I used up my time. So let me summarize. The story of bacteria is done by 2 of my students. Xin Wang, he did all the theory, and Xia Kang did all the experiments. It's published recently. And stem cell story is in collaboration with my colleague Honghui Deng. And the theory part is done by my student Ziyuan Li. She's now a faculty member at Peking University. And then this DNA model is done by Jinxiang Shen, also my student. And we just submitted the work. Okay, thank you.
Let's take time for a couple more questions.
it comes to identifiability in dynamic systems, there are a lot of fundamental theorems that don't include the mapping or the estimator, right? So when I saw this, it seemed like it was working very well, this neural network-based way of identifying these dynamic systems. But it can't generally subvert these identifiability problems. So are there assumptions about biological dynamic systems built into what you're doing that make this so effective in the examples you showed us?
Yeah, very good question. So we try to, we ask the same question as you did, right? And then we say how this method can infer network for arbitrary topology. It turned out that the performance is much worse, as you might have guessed. Okay, you will either overfit or underfit. You have to really choose the training data and the prediction data and all this. You can do it, but not so robustly. And here you can do very robustly. We believe that this has something to do with the robustness of biological networks. Yeah.
So a biologist would say that A activates C versus A inhibits B inhibits C are two different things. What happens if you do the double mutation experiment? If you knock out B, does A still activate C?
What if I do a double? I don't quite understand your.
So if A were only acting on C by way of B,
Yeah.
then if you take B out of the system,
Yeah.
A would have no effect on C.
Right.
So you can distinguish between your model.
Okay. I now understand. Yes, you asked me that. Can I really refine the model by performing more mutations? Yes. Yeah. If we put more training data, because that's kind of cheating, right? Yeah, we can do. Yeah, we can do that. Yeah, but here we want to use the minimum training data, which is just 7 frames of the Y-type dynamics, and to predict hundreds of mutants. Yeah.
Let's thank Chao for a great talk.
Okay. Thank you.